So um, last lectures we talked about mitotic division. So this is asexual, and you produce an exact copy of the mother cell. There's some advantages. It's really fast. Bacteria can reproduce every 20 minutes. And so you can take some, put it in a dish, and or put it in culture, and the next day it'll be completely full to saturation. Um, there's an issue though, which is that since you're making an exact copy, then you have poor adaptability. So you can't adjust to new environment. So if you have something in which you've grown in, let's say 37 degrees Celsius, and then for whatever reason, temperature drops to 30 degrees Celsius, right? You cannot adapt to that new environment um, because you're making identical copies of yourself with the exception of limited um, mutation rate. So sexual reproduction allows, I can't spell, allows for genetic variety. And this is really a profound um, thing, right? I mean, in sexual reproduction, at least in animals, we have a situation in which one half of the species cannot um, propagate. All right, so the males of the species, they donate a little bit of DNA, but they don't have reproductive potential on themselves, right? That's a huge waste of resources, as are men in general, right? They're just consuming and consuming, and then, as in life, the women do all the work, right? So the advantages to sexual reproduction must be greater than the cost of having 50% of the population not actually actively being able to reproduce. And we'll see what those advantages are going forward. So we've already talked about, brighten this up, um, asexual reproducing organisms. Here's a little bacteria. And then here in sexual reproduction, the key thing is that the offspring are genetically different from the parents. And the way in which that occurs, and this occurs in which every organism that undergoes sexual reproduction, is this process called meiosis, which has gametes, which are not functional cells, but they can contribute to form new cells. And those gametes are genetically different from the parents and also from each other. And these gametes will have one half, oops, the DNA content of the original parent cell. Let's go blue. So um, here, the mechanism in which genetic variation occurs is through a couple of things, and we'll see each of these, and we'll see when they occur during meiosis. One is crossing over or homologous recombination, and this is where there is a literal physical exchange, and you see this dark green here, physical exchange of DNA between the mother and the um, father cell. And so this is interesting. The chromosomes that you have in your own body are not directly from your mom or your dad. It's not exactly identical to your mom and dad's. It's actually a combination of your parents, grandparents' chromosomes that have all been mixed up during the meiotic process, and then you get them. So one, you have this direct physical exchange of the DNA, and then you have the random assortment of homologous chromosomes. And so we'll see in meiosis that if you're a diploid, you have two in DNA content, two pairs of chromosomes, you will only inherit one. And whether that one you inherit from your dad, it could be either his dad's or his mother's chromosome. And so there's some variation there simply based on the random assortment of the chromosomes. And then you are, unlike in mitosis, you're actually getting genetic material from two individuals instead of just the one parent. So all of those increases genetic variation, and we'll see that we can go to basically an unlimited amount of variation um, within by the mitotic process. And these are the sex chromosomes here, X and Y, and they play a key role in determining gender and variation. Now, this is how it works in humans, but every different organisms have different ways to determine gender, for instance, or reproduction potential. So here, um, the we have an X and Y. In wasp and bees, it's the number of chromosomes. So females have two chromosomes and males only have one. Here, in this little turtle, the females are born at a higher temperature. It drives towards females, and at lower temperature, it drives towards male. For us, as humans and for animals, um, it's determined by our sex chromosome. So men are XY, females are XX. There is a random division of these chromosomes during meiosis. So females will always have either X or X. Men, when these chromosomes segregate, they will deliver either an X or a Y. And so when fertilization occurs, the female will always provide the X chromosome, and then the man will provide either the X or the Y chromosome. If you're XY, biologically male, XX, female. Um, And so obviously, I don't think I need to say this, but biological sex is different from gender. Gender is kind of a, mental construct, and so there is some separation between gender and sex. But this is driven by the chromosomes. We'll see later that there are all sorts of situations that occur, right, that are very confusing. I mean, there could be non-disjunction events that give an XXY. Um, or something to that effect. And then with this individual, well, you know, uh, um, biological sexes or gender are relative things. So this process only occurs in sex cells. Our somatic cells, or also called our autosomal cells, these are body cells and they don't participate in reproduction. So these are most of the cells in our body, right? And they're just living their life. They will reproduce by mitosis almost exclusively. These somatic cells have pairs of chromosomes, right? And so we've seen that in the karyotype, they'll have two uh, uh, chromosome number ones. And then remember in karyotype, they're organized by shape and size. So they have two chromosomes number two, and oops, my shapes are out. And two pairs of chromosome number three. One of these are from mom, and one of these are from dad, right? And there's no kind of relation. This one, dad, mom. Now, what's important in mitosis is that when all the mitotic processes happen, these chromosomes don't associate with each other when they segregate. So in mitosis, chromosome segregation is independent of its homologue. In meiosis, we will see that proper chromosome segregation is absolutely dependent on a homologue, and they have to come together before chromosome segregation can occur. And we'll see during mitosis, you really don't need your homologue at all because what? In mitosis, the segregation occurs at the centromere. They're pulled on either side of the centromere, and so the sister chromatids will segregate fine. It doesn't matter what's going on with its homologous chromosome. Here's a karyotype right here. And so you see these are homologous chromosomes of chromosome 1. And you see that all of the chromosomes, this is in humans, have a homologous chromosome. We know this is a female because this person has two X chromosomes. If it were male, you'd have one X chromosome, and then the Y chromosome is just this like little short, stubby little thing that is very small. Only a couple of genes on the Y chromosome, they just happen to be the ones that um, um, drive uh, male processes. And this is a look at a diploid organism, at least for an animal. Here, most of our life is as a diploid. So as soon as fertilization occurs, we basically live as a diploid animal. But in our sex cells, they undergo mitotic division to give rise to haploid uh, um, chromosome numbers that allow for propagation. So this will be the sperm and egg in humans and in animals. So note the number here in here allows us to generalize for the chromosome number. So a haploid number is the number of chromosomes, and then diploid would be the number of pairs. So in humans, we have 23 chromosome pairs and 46 chromosomes total, right? So 46 is our diploid number. This will be associated with 2n, and 23 is the haploid number, which is associated with n. And so we, as part of our life cycle, are haploid only in the sex cells, and then most of our life is diploid. That's not the case in some organisms like yeast. Um, if we look, the yeast life cycle, they can be kind of 50-50 haploid and diploid. So they can grow, they can become diploid, they can reproduce asexually, and then undergo spor sporulation to become haploids, or they can actually grow as haploids as well. So there are differences depending on the organism. Some plants have different organisms as well. And this is a look, obviously, we're going to go down in detail, but I'll introduce just what's happening in meiosis here, and then I think in the next lecture we'll go into more detail. So here, meiosis is two divisions. So there's a meiosis 1 and a meiosis 2. We will see meiosis 2 looks like mitosis in many ways. And meiosis 1 is different. We call this a reductional segregation. Because here, meiosis 1, we go from two pairs of one pair of homologous chromosome, one pair of homologous chromosomes, stop that, to one individual homologous chromosome. So, in meiosis one, you have a prophase. This prophase, just like in mitosis, involves chromosome condensation, but there's a lot of special stuff that happens during prophase of meiosis one. In females, meiosis one lasts for the life of the organism, and humans, and kind of only completes during ovulation. Um, uh, but we'll see that in a little bit. So here we have condensation of the chromosomes. Stop that. And prophase one is divided up in all these phases. I'm not going to make you learn, but there's like diploquine, diakinesis. Um, uh, I got my PhD in this. I should remember all of these, but um, zygotine and whatever. Hey. Okay, so in prophase one, you have condensation of the chromosomes. Now, what happens in meiosis one that is unique is that the homologous chromosomes, stop, find each other. And so here, these are combinations of homologous chromosomes which are finding each other and are beginning to wrap around each other. And so you can see this in this cartoon here. You have the homologous chromosomes. It's one from mom, one from dad. They find each other and begin to wrap around each other. And this is where recombination occurs, or crossing over occurs between those strands. And we'll go into more detail about that. And so in late prophase, these homologous chromosomes found each other and undergone a process called synapses, in which the DNA actually exchanges. And this is a look at this. This is early prophase, sorry, interphase. Uh, this is the first part of prophase, which is leptotene. So we're beginning to see chromosomal condensation. You're going to see it at the edges here, but homologous chromosomes are just forming their initial contacts with each other. In zygotene, you see that the chromosome becomes denser and the homologous chromosomes have begun to find each other. Pachytene, it's kind of like they're packing together, so they become denser still. And then diplotene is where you can see recombination occurring. So this is a homologous chromosome pair here, and you can see there's a region of crossing over there and a region of crossing over here that will be the sites of recombination um, uh, and DNA exchange in these chromosomes. And then in diakinesis, they have completed this synapses and are ready for um, mitosis one. Um, so this is the crossing over that we see in diakinesis and in pachytene. Oops, and what's the last one? Oh, diakinesis and diplotene. Um, And then these are where the exchange of genetic material occurs. So the result here is that the homologous chromosomes are going to create uh, chromosomes in the gametes that are different from the ones that originated. And so let's say you have chromosome 1 from mom here and chromosome 1 from dad here. If a recombination event occurs, you end up with chromosomes. So remember, these are actually two chromatids, right? So instead of ending up with chromosomes that are exactly the same as mom and dad's, you end up with one that looks, something like this, where these chromosomes are now combinations of both your maternal and paternal chromosomes. So these are different. Now, remember this, right? We said before that chromosomes, homologous chromosomes, they have the genes in the same location on the same chromosome. So if there's a gene, I'm running out of colors here. If there's a gene for insulin here, that gene will be located at the same place in both mom and dad's chromosome. Now, the individual gene might be different. This one might produce, super insulin or whatever, whereas this one might produce lackluster insulin, right? So in the gross level, in the kind of large picture, the genes are going to be the same, but on an individual level, there might be differences. We call those differences alleles, and we'll get into that in actually Biology 112. And so going over here to our recombinant chromosomes, we would still have those genes in the same place, right? And so in this particular case, this insulin gene would be kind of in the paternal, this one's in the maternal, but look down here, if you have another gene here, right? And let's say, make this like a slightly different blue over there. So these are different genes. They're the same genes, but they are different alleles of those genes. And now you have one over here and a different one over here. So between the two of them, this combination and this combination did not exist in the original parents, right? So this is a what? Recombination of these genes, these different genes on these chromosomes, right? And so what this allows is not the creation of new genes, but recombines the pairing of different genes to allow for different interactions. So it could very well be that a combination AB over here might allow for super insulin production, whereas a combination of, AB over here, these alleles might allow for lackluster insulin production. And so this is a way to increase the variation simply by mixing things up. Ooh, okay, so that was a long explanation, but I'll finish up meiosis 1 and then we'll talk about meiosis 2 on the next lecture. So this is a look at the recombination event. So here is one homologous chromosome, and these have already undergone DNA replication. So this is one chromatid. This is another chromatid. This will be from mom, let's say. And then here we have one chromatid and one chromatid from dad. And the recombination occurs between individual chromatids. So the recombination would be between this one and this one. This particular chromatid might be left alone. And so the result is that you would have, what would actually happen is that this one would kind of recombine with that one and all sorts of things. The result is that you will have four different unique chromosomes, none of which that look like the original parent chromosomes that came about. These areas of crossing over here and here, they are called chiasmata. Chiasmata, I think that's Latin for X or something to do with X, chiasma. And so those are the points of crossing over. And I actually spent my PhD dissertation looking at recombination events in meiosis and determining how they work and how we can alter those processes. So when that happens after prophase one, then the homologous chromosomes align along the metaphase plate. Stop that. And in anaphase one, the homologous chromosomes segregate from each other. And so now where we had a pair of chromosomes, you only have one chromosome. And so from here, we've gone from um, a um, and here we've gone from pairs, stop that, pairs of chromosomes to individual chromosomes. In telophase one, there's a little bit of relaxation that occurs, and then you go through another round of division. And this is a round in which the sister chromatids segregate from each other. And so the sister chromatids segregating from each other looks like mitosis. So you have prophase one, condensation. You have metaphase one, where they're aligning along the metaphase plate. I'm sorry, two, metaphase two. Anaphase two, where the sister chromatids segregate from each other. Telophase two, and then the products. And so the resulting products here are four haploid products from uh, one diploid parent. Um, and so here we have meiosis. So in meiosis, you start with a diploid parent. So note this is just one that has two chromosomes here, right? One, two, one, two. So two pair of chromosomes, four chromosomes, that's 2N, right? There's a round of DNA replication to form sister chromatids. And then in prophase one, the homologous chromosomes find each other and they undergo recombination. In metaphase one, the homologous chromosomes segregate from each other. So note the blue is segregating from the red here. So they are segregating and going to different poles. Now, during anaphase one, this is where the recombinant chromosomes segregate from each other as well. You can kind of see here that when they're pulled by their centromeres, and you see that this little piece of blue is coming off with the red here, and this little piece of red is coming off with the blue there. So the recombinant chromosomes are segregating from each other, and the homologous pairs are segregating from each other. Telophase one, um, you have a kind of reformation of nuclear envelope, and then the cell sets up again to undergo another round of replication. So metaphase two, in which the sister chromatids segregate from each other, and then the formation of the haploids after telophase two. Whew, that is a lot. Um, So let me stop and then I'll talk about some non-disjunction events and what goes on in humans.